Hello everyone and welcome back to AP Bio with AP Playground. Today we're talking about topic 1.7, proteins. Lots of objectives today, but all boiling down to describe the structure and function of proteins. So we're going to look at the formation of individual amino acids, the parts that make them all alike and the parts that make them different, as well as the different levels of protein folding that create a protein structure overall, as well as the function of proteins. And suggested skill is argumentation, 6E, predict the causes or effects of a change in or disruption to one or more components in a biological system. Just like all of our other macromolecules, we have to take a look at the monomers and polymers of proteins. And remember, monomers are the building blocks, while polymers are what's going to be built. And proteins are made up of CHON, meaning the essential elements that they contain are going to be carbon, hydrogen, oxygen, and nitrogen. So proteins are the polymers made up of amino acid monomers. Those are the building blocks. And every single amino acid has these elements. There's going to be a central carbon, as we can see here, an amino group, which you should know from our lesson 1.2. An amino group is going to be a nitrogen bound to hydrogen atoms, a carboxyl group, a carbon double bound to an oxygen and bound to a hydroxyl group, a hydrogen atom, and then an R side chain. And the R side chain is going to differ from amino acid to amino acid. And so amino acids are linked by peptide bonds, forming polypeptides, otherwise known as proteins. And so we're going to be taking a look at how peptide bonding occurs, but first let's delve deeper into the R side chains. Now, the R side chains, or the R side groups of amino acids, can be polar slash nonpolar, so they're charged or not charged, and also basic or acidic. They can give up hydrogen atoms, or they can accept hydrogen atoms. So when we're looking at the different properties of side chains, we're looking for one, are there ions? That's going to make them charged. Two, electronegative elements. Is there going to be polarity? And three, do we have functional groups that could be acidic or basic? And so these properties are going to affect the structure and function of the protein. And of course, all of these amino acids are going to have the same backbone, but the side groups are going to be different. So looking at this first side group, we just have a carbon ring, carbons double bound to each other, no ions, no electronegative elements, no functional groups. This is just going to be a non-polar amino acid. Now over here, we also have some hydrocarbons, but we're also looking at this ion here, this amino group, and it's going to be basic. It can give up a hydrogen ion, it can give up a proton. So this is going to be both ionized and basic, and it has a charge. And that's going to be especially important when we're looking at proteins within aqueous solutions. Are they going to be polar or nonpolar? Are they going to be hydrophilic or hydrophobic? Now over here we have another group, another negative ion, an anion over here, and because of this oxygen without a hydrogen atom, but it's an oxygen with an extra electron, it can accept a proton, a hydrogen ion. So this is going to be ionized and acidic, and again it's going to have a charge. And lastly, over here, we have a few methyl groups. So we are looking at functional groups here, but it's just carbons bound to hydrogen atoms. So they're not going to be ionized, they're not electronegative. So this again is a non-polar amino acid. And now we get to the most complex part of protein structure, protein folding. And proteins have 4 levels of structure, definitely more than the other macromolecules. And the primary structure is just going to be your sequence of amino acids. So just each individual amino acid with its side chain bound to each other. So if this is amino acid 1, it's going to have a peptide bond to amino acid 2, peptide bond to amino acid 3, and so forth and so forth. And then we have our secondary structure, where the segments of the polypeptide chain, and this is the polypeptide chain we've created right up here, is going to be coiled or folded due to hydrogen bonds formed between the backbone. So we're not even looking at the side groups yet, but the backbone of each amino acid has the potential to form hydrogen bonds. And so these hydrogen bonds can either form an alpha helix, as we see here, we're looking at a helix, or a beta pleated sheet. where this is going to be obviously more of a sheet shape than a helix. And again, this is backbones. These are bonds formed between the carboxyl, the amino, the hydrogen within the backbone, and not based on the R side chain. And let's continue on. Now we have tertiary structure. So now we're going to be taking a look at the side chains, or the R chains. And this is going to be folding of the polypeptide chain due to interactions between the side chains. So over here you can see that all of a sudden it got a lot more squiggly because of the interactions between these various charged and non-charged. These various ions are bonding. There's going to be just sorts of different bonds, as well as hydrophilic and hydrophobic side chains. Especially if the protein, like I mentioned earlier, would be near an aqueous solution, then all of the hydrophilic side chains would try to get their amino acid to be closer to the outside of the protein, while the hydrophobic side chains would be closer to the inside of the protein. And as a result, we get quite a wild mumbo-jumbo shape within tertiary structure. And so we have ionic bonds, disulfide bridges, hydrogen bonds, and hydrophobic interactions, all of which we'll discuss closer in the next slide. And then we have quaternary structure, and this is when we have multiple polypeptides, so that have already gone through the first 3 levels of protein structure, attaching to each other using the same bonds that we see up here. So they're hydrogen bonding to each other, they're ionically bonding to each other. We're seeing disulfide bridges between multiple polypeptide chains, creating a much larger protein consisting of multiple polypeptide chains. Now, not all proteins get to this level. Some remain one polypeptide chain, but this is a possibility. So now let's take a closer look at tertiary structure. So we know about ionic bonds, a bond between a positive and a negative ion. We know about hydrogen bonds due to the electronegativity of oxygen causing a partially positive charge, and then a partially negative charge on the oxygen, creating a hydrogen bond. But we also have some new bonding that we haven't talked about yet. There are disulfide bridges, which are bonds between disulfide groups, or 2 sulfhydryl groups, so 2 sulfur atoms. We have hydrophobic interactions, and these are formed when hydrophobic side groups are folded so tightly that van der Waals forces form. So remember when we discussed being in an aqueous solution, all of the hydrophilic side chains and amino acids are going to be on the outside, while the hydrophobic ones are going to be pressed tightly on the inside, trying to get away from the aqueous solution, and they're going to be pressed so closely together that we're going to see hydrophobic interactions. And so van der Waals forces are when 2 atoms are so close to each other that the random movement of electrons around the atoms could result in charged sides, which attracts the 2 atoms. So, of course, within an atom, we have electrons that are moving around, and by random chance, there's a possibility that one side of the atom, or the electron cloud around the atom, would have more electrons in it than the other side. And that's going to result in very unstable, very slightly positive and slightly negative charges. And then this happens within another atom that's really close by, and we can create a very weak bond. So looking at protein folding as a whole, you can see that a change in primary structure can greatly affect the rest of the protein, because a change in the primary structure could possibly affect whether we're having an alpha helix or beta pleated sheet. Then it's going to affect the tertiary structure and polypeptide chain, because even one change in amino acid is going to affect the folding of the chain, depending on what kinds of bonds can be formed between this new amino acid and the other amino acids around it. And as a result, this is going to change the structure, which in turn will affect the function of the protein. And additionally, when secondary structure and tertiary structure bonds are broken, this can either be due to pH that is not at an optimal level, salt concentration that is not at an optimal level, or temperature that is too high or too low. The protein is going to unfold in a process known as denaturation. So in order to keep a protein intact, it is also key to keep it in an environment, to have it in an environment in which the secondary and tertiary structure bonds can hold. Proteins are going to have various functions, including acting as catalysts in chemical reactions, so helping chemical reactions occur with less energy spent. They store amino acids. We're coordinating an organism. We're helping with movement and muscles, defense of the organism and the immune system, such as antibodies. We've got transport of things between cells, as well as transporting signals between cells that can then be recepted by proteins, allowing for the transport of items through the cell membrane. We have support, structural support such as collagen and keratin. We have building and repairing parts of the body. And lastly, we have expressing genetic information. And throughout the rest of this course, we'll be covering a lot of these different functions of proteins. So it's good to have a list here. So there you have it. Proteins are made up of amino acids that have different side chains. These interact within protein folding of the four structures, primary, secondary, tertiary, and quaternary, and proteins have a lot of functions ranging from movement to the immune system to cell transport.